Hello everyone. Today we shall look into colorimetry and spectrophotometry, also good laboratory practices, handling of poisons and hazardous chemicals. So let's first look into colorimetry. Colorimeter refers to a device that is used in colorimetry that aids in the absorption of a particular wavelength of light by a specific sample solution. It was invented by Louis J. Dubček in the year 1870. Why we need this colorimeter is to measure how much light it transmits and absorbs as it passes through a liquid. Then this actually colorimeter is used to identify the color and establish the concentration of a solution. Now, by comparing solute's color intensity in a solution to that of a reference solution with a non-solute concentration, one can estimate the concentration of color solute in that solution. Then when coming into the principle of colorimeter, when an incident light beam with intensity I0 passes through a solution, a part of the incident light is reflected, IR, and absorbed IA, while the remaining incident light is transmitted. So it can be compiled and written in an equation that is I0 is equal to IR plus IA plus IT. The measurement of I0, that is the incident light in the colorimeter, eliminates IR, that is the reflected light, and it is sufficient to calculate the IA, that is absorbed. Using the cells with the same characteristics maintains a steady amount of light reflection. Then IO and IT are measured. Then the colorimeter follows the principle of Beer-Lambert law, which states that the amount of light absorbed by a color solution is directly proportional to the solution's concentration, the length of a light passed through it. That is, absorbance is directly proportional to C and L. Absorbance, to eliminate the proportionality sign, we put coefficient absorption ECL. Okay, where A is absorbance or optical density of solution, is coefficient of absorption, C is concentration of solution, and L is equal to length of the path. Now, in a colorimeter, we have a series of lenses, okay, which guide a beam of light with a particular wavelength through solution as it makes its way to the measuring apparatus. This compares solution color to a current channel to examine it. The absorbance or percentage transmittance is then calculated using a microprocessor. By measuring the difference between the amount of light at its source and that after passing solution, it is possible to determine the concentration of solution and how much light will be absorbed. Then path to the colorimeter, we have a on or off switch control, then a sample holder, that is cuvette, then wavelength readout, absorbance readout, then mode indicators, mode select button, decrease increase wavelength control, 100% transmittance control. Then colorimeter examples: colorimeter CLRS bioevapic, then water analysis colorimeter checker, then photoplex STD colorimeter, laboratory colorimeter. So this is the example of a colorimeter where light source and monochromator, cuvette, sample, amplifier, and output. Several sample solutions with the known concentrations are first prepared and evaluate setting the concentration of an unknown sample. So we have a sample solutions with the known concentrations, then they are evaluated so that we can know the concentration of an unknown sample. Plotting the concentration versus absorbance on a graph yields calibration curve. So when we plot a graph where it is concentration on the x-axis and absorbance on the y-axis, yields a calibration curve. To determine the concentration, results of the unknown sample are contrasted with those of the known sample. So to know our unknown sample's concentration, what we are going to do is we are going to contrast with the result that we have got for the known sample curve. Then instrumentation of colorimeter. What are the parts present in a colorimeter? First and foremost, is the important one is light source. The source of light should produce energy with enough intensity to cover the entire visible spectrum. Commonly, tungsten lamps are used as a light source for measurement in the visible spectrum and near infrared ranges. Halogen deuterium is suitable for measurement in the UV range. Next one is slit. Why we need a slit is to reduce the unwanted light beam to pass through. Then we have a lens, condensing lens. which parallel beam of light emits from a condensing lens after the light passes through slit incidence on it. So first light source passes through the slit, then to the condensing lens where parallel beam of light is passed through this lens. Next is monochromator. Monochromator is a filter which it filters the monochromatic light from polychromatic light, which absorbs unwanted light wavelength and permits only monochromatic light. That is, monochromator acts as a filter which absorbs unwanted wavelength and only permits whatever wavelength we need. That is, monochromatic light is passed through it. There are three types. Monochromators are three types: prism, grating, and glass. Then prism. Monochromator one, prism. What it does is it facilitates the refraction of light when it passes from one medium to another. Glass is selectively transmits light in certain range of a wavelength. Gratings are the ones made of graphite which separate light in different wavelengths. Then cuvette. Cuvette is a sample set. Monochromatic light from the filter passes through the colored sample solution placed in the cuvette. So the light from the filter is passing through the cuvette. Cuvette has a sample solution. Then their sizes range from square, rectangle to round and have a fixed diameter of 1 centimeter. So the cuvette can have a shape of square and rectangle and has a fixed diameter 1 centimeter. Then there are three types of cuvettes based on the substances, that is glass, quartz and plastic cuvette. Glass cuvettes are cheap and they are absorbing the light length of 340 nanometer wavelength. Then quartz cuvettes are with entry of both light UV and visible ranges. Then plastic cuvettes are cheaper, but what happens is they get easily scratched and they have short lifespan. Then photocell. Photocell is devices which measure light intensity by converting light energy into electrical energy. Then galvanometer, it is electrical signal generated in a photocell is detected and measured by a galvanometer. It displays optical density and percentage transmission. Then types of colorimeter. Densitometers, which determines the density of a material. Then spectrophotometer, which is mostly used in the laboratories. It measures the spectral reflectance and transmittance of a surface. Then tristimulus colorimeter employed to measure the tristimulus, that is, measure the light intensity of the primary color values in a sample. Then where we apply this colorimeter, it is employed in the printing industries to evaluate the caliber of printing ink and paper. Then food and food processing industries, and also it is frequently used in the laboratories and hospitals to determine the biochemical composition of samples like blood, urine, cerebral spinal fluid, plasma, serum, etc. Then it is used in textile and paint industries. Diamond dealers also need this colorimeter for visual characteristics of priceless stones using a colorimeter. Then they use it to evaluate the water's purity and screen for the presence of chemicals like cyanide, iron, fluorine, chlorine, molybdenum, etc. They are employed to evaluate the color, contrast and brightness of screens on mobile devices, computers and televisions to give people the greatest viewing experience. Next is advantages of colorimeter. So first one is a quick and affordable means of evaluating quality is a colorimeter. Then it makes it simple to perform a quantitative examination of colored chemicals. Results are available in under a second. Then 4 AA batteries can be measured by using a portable colorimeter between 100 and 300 times. Then if anything has advantage, it will be having disadvantage. Then what are the disadvantages? That is concentration of a substance. When we are measuring through with the help of this colorimeter, it become a laborious process. Then colorimeter does not function in the ultraviolet or infrared spectrum, since it only measures wavelength absorbance in the visible range of length. So when it comes to colorimeter, we can only check in the spectrum in visible length. Then next is a spectrum range must be set rather than specific wavelength to measure the absorbance. We have to set a spectrum range. We cannot set a specific wavelength. The measurements might be if the thing is not clear, I'm really sorry. Then next is spectrophotometer. What is spectrophotometer? It is an instrument that measures the amount of light absorbed by a sample. Then spectrophotometer techniques are mostly used to measure the concentration of solutes in a solution by measuring the amount of the light that is absorbed by solution in a cuvette placed in the spectrophotometer. Scientist Arnold J. Beckman and his colleagues at the National Technologies Laboratory invented the Beckman-Dewey spectrophotometer in 1940. Principle of spectrophotometer. Spectrophotometer technique is used to measure light intensity as a function of wavelength. It does this by refracting the light beam into a spectrum of wavelength, detecting the intensities of the change coupled device, and displaying the result as a graph on the detector and then on the display device. In the spectrophotometer, a prism or grating is used to split the incident beam into different wavelengths. By suitable mechanisms, the waves of specific wavelengths can be manipulated to fall on the test solution. The range of the wavelength of the incident light can be as low as 1 to 2 nanometer. The spectrophotometer is useful for measuring the absorption spectrum of a compound that is absorbed by light by a solution at each wavelength. Now, instrument. Essential components of spectrometer instrumentation include energy source, then a monochromator, prisms, gratings. So before that, energy source, materials that can be excited to a high energy state by a high voltage electric discharge or by electrical heating serve as external energy sources. A monochromator to break the polychromatic radiation into component wavelength or band of wavelength. A monochromator resolves polychromatic radiation into its individual wavelength and isolates these wavelengths into very narrow bands. Prisms. A prism disperses polychromatic light from the source into its constant wavelength by virtue of its ability to reflect different wavelengths to a different extent. We have two types of prisms which are employed here, namely 600 corner quartz prism and 300 litro prism. Within gratings, gratings are often used in the monochromators or spectrophotometers operating ultraviolet, visible and infrared regions. Then transport vessels or cuvettes to hold the sample. The sample which has to be studied in the ultraviolet or visible region are put in the cuvettes. Then cuvettes meant for the visible region are made up of ordinary glass or sometimes quartz. The next one is a photosensitive detector and an associated readout system. Most detectors depend on the photoelectric effect. The current is then proportional to the light intensity and therefore a measure of it. Radiation detectors generate electronic signals which are proportional to the transmitted light. These signals need to be translated into a form that is easy to interpret. This is accomplished by using amplifiers, ammeters, potentiometers, and potentiometric recorders. Application of spectrophotometers. They are used in the detection of concentration of substances, detection of impurities. Then they are used to elucidate the structure of organic compounds. Then for dissolved oxygen content in freshwater and marine ecosystems, we can use spectrophotometer. For protein characterization, we can use spectrophotometer. Detection of functional groups, respiratory gases analysis in hospitals. Then for knowing the molecular weight of compounds. Then the visible and UV spectrometer can be used to identify class of compounds in both the purity and biological preparations. Next is good laboratory practices. What is GLP or good laboratory practices? It is a quality system guideline covering the organizational process and conditions under which non-clinical laboratory studies are planned, performed, monitored, recorded, reported, and archived. GLP ensures the quality and integrity of a safety test data submitted to the government for the issuance of research permits. Then GLP or good laboratory practices are a set of quality assurance principles that ensures the reliability and consistency of a laboratory operations. When dealing with the poisons and hazardous chemicals, strict adherence to the GLP is paramount to protect the personal, the environment, integrity of experimental data. So, what are the principles for handling poisonous and hazardous chemicals? First, we have to assess the risk. So, we have to identify potential hazards associated with each chemical. Then, assess the risk involved in handling, storage, and disposal. Then, develop appropriate safety measures based on the assessment. Let's come into PPE, personal protective equipment. Wear appropriate PPE, including lab coats, gloves, safety glasses, and face shields as needed. Select PPE based on the specific hazards of the chemicals being handled. Safe handling process. Avoid generating aerosols or dust. Use proper techniques for transferring, mixing, and dispensing chemicals. Work in well-ventilated areas or use fume hoods when necessary. Never pipette by mouth. Emergency preparedness. Have emergency equipment readily available such as eye wash stations and safety showers. Develop and practice emergency response plans. Ensure all personnel are trained in emergency procedures. Chemical storage. Store chemicals in appropriate containers and cabinets. Label containers clearly and accurately. Separate incompatible chemicals. Store flammable and reactive chemicals in designated areas. Waste management. How to manage the waste that is there in the laboratory. We have to collect the waste properly, then have to segregate it and dispose of chemical waste. Comply with all environmental regulations. When disposing the waste, it should not contaminate the environment. Training and education. Provide comprehensive training to all laboratory personnel on chemical hazards, safe handling procedure, emergency response. So, to every laboratory person who is handling with these chemicals, we have to give them a training on chemical hazards, how to handle it safely, and also how to respond when there is an emergency. Then conduct regular safety training and refresher courses. Documentation. So we have to document all the details of these chemicals, handling procedures, accidents and waste disposal. Then we have to establish documentation protocols. Specific precautions for poisons. Handle poisons with extreme caution. Then poison handling should be done in designated areas only. Limit access to poisons. Then implement strict controls for the disposal of poisonous waste. Additional considerations. Chemical compatibility. Understand the compatibility of chemicals to prevent hazardous reaction. A laboratory must be ventilated properly. So ventilation, ensure adequate ventilation to control exposure to chemical fumes and vapors. Spill response. When there is a chemical spill, we have to have a plan for responding to such chemical spills. Then regular inspection. We have to conduct regular inspections for the laboratory to identify and address safety hazards. Compliance with the regulations, adhere to all the relevant local, state and federal regulations. By following these GLP principles, laboratory can significantly reduce the risk of accidents, injuries, and environmental contamination associated with the handling of poisons and hazardous chemicals. So, this is what I have to say. Thank you.